Hi, my name's Scott Berry, a biostatistician here at Berry Consultants, and I'm going to talk to you today about sharing controls and platform trials. In order to introduce you to platform trials, let's think about what current development looks like. So this is a standard trial that's run now, and I'll refer to this as solo trial development. There's maybe up to 2 years of planning the trial. You create the sites for the trials, you create a protocol, you visit regulators, you get IRB approval, you get all your infrastructure set up, and then you run a trial, and it's probably a 1-to-1 randomized trial, active to placebo. And then there's a process of closing down the trial, closing down the sites, and the trial's over. And this is what a standard clinical trial looks like. If you think about it at a larger level, the development of different trials being run in the same disease. Here's an example of 6 different trials being run in the same disease, everybody running 1-to-1 randomized trials versus placebo within the disease. And we know how we got to this solo development, parallel development of individual companies. Here's an example of the ramifications of the solo development. You can see on the right the 6 different agents being run separately in their trials. First of all, there's a lot of white space. There's a lot of planning and shutting down for all of the different trials. We've reproduced the same thing 6 times. They're separate inferential units as well. None of the data from the first trial contributes to the inferences from any of the other trials. We have different protocols, inclusion-exclusion criteria, different visit schedules. These are separate inferential units. Notice 50% of all patients enrolled across these trials are placebo. And we're enrolling too many placebos for the inferences in these arms. We enroll 1-to-1 in each one of them for increasing the power of each one of them because they're separate inferential units. A platform trial, and my slide here is showing you an arena, a standing infrastructure where multiple treatments come into the same trial. So a platform trial is one where multiple investigational agents or treatments are explored in the same trial. The same protocol, we refer to this as a master protocol. The master protocol will have appendices for drug A, drug B, drug C within that trial, but it's the same trial. And patients will be randomized to the different experimental arms in this trial. We are now constructing one trial. We are bringing multiple agents into that. We're reducing white space. We're sharing infrastructure, sharing costs. And the discussion of this particular topic is sharing control patients, which is a huge inferential advantage for doing this in the same trial. So let's look at what that looks like. Here again are 3 separate trials for red, green, and yellow. Each one of them individually enrolling 90 patients on active, 90 on placebo, and let's consider them as having 90% power in their individual trial. Globally, we enroll 540 patients, 270 on active and 270 on placebo. That's what the global look at the separate trials is. Now let's look at a single platform trial where the same drugs are enrolled, but we enroll 90 to the active arm and 30 to the placebo for that arm. So you can see we have 90 on red and 30 on red's placebo. They're enrolled in the same trial, so when each of the individual active agents we make inference, we pool together and combine the placebos so we have 90 placebos. Red is being compared to 90 placebos, green to 90 placebos, and yellow to 90 placebos. But we only enroll 90 globally rather than 270. Each arm has the same 90% power. Globally, we're enrolling 360. So we have a 33% reduction in enrollment. we have a 67% reduction in placebo. So only in this setting, we're enrolling 3 to 1 individually, so we enroll 25% placebos rather than 50% placebos. Each particular arm is shorter to enroll, enrolls less patients, and the sharing of infrastructure. But for now, we'll focus on the sharing of placebos. So you can see the huge advantage of platform trials in resources with the same or actually increased inferential ability for less resources. Now, these platform trials are a relatively new innovation, but they're being used in a wide variety of therapeutic areas. A number of oncology scenarios are being done, from I-SPY 2, glioblastoma, neoadjuvant breast cancer, but it's being done in a wide variety of infectious diseases. It's being done in Alzheimer's, Crohn's, pain, wide variety of therapeutic areas. I'll touch on the ALS in a couple slides as well. There's a new IMI2 initiative that's looking at integrated research platforms, and they're looking at platform trials within 3 separate diseases as well. So this is an advancing science being done in a wide variety of areas, and the sharing of controls is a huge potential benefit of these trials. Here's a publication that I was involved in with Ben Seville that looked at adding innovative features to these platforms as well. I won't go into the details of this. You can certainly look at this publication, but it looks at adding perpetual platforms. So rather than a finite time platform that investigates 3 drugs, we just keep adding drugs continuously throughout the course of the trial as the need exists. You can drop arms that are demonstrating that they're not doing well, drop them for futility. You can do response adaptive randomization across the arms, and so it increases the number of patients enrolled to more effective arms. You can extend arms to larger sample sizes as well. So this particular publication looks at these advantages within the design. And I'll just touch on the high-level aspects of doing separate solo development to find a positive arm. Can be as many as 2,000 patients. That can be reduced all the way down to 640 patients on average by adding these innovative features of perpetual platform and response adaptive randomization. So if you'd like to read more about those innovations, you can look within the paper for adding innovations to these platforms. Here's a slide that was presented by Mass General Hospital, the Healy Center for ALS, on a platform trial they're running in ALS. And they presented the savings through the platform, the time to find a therapy that demonstrates efficacy, reducing time from 8.5 years to 3.4 years, and the number of subjects needed from 1,800 to 880. So huge benefits from the traditional approach to using these platforms, and the sharing of controls is a huge part of these. Now, a number of statistical issues come about when you're modeling control and you have these controls in these platform trials. One of them is I showed you an example with red, yellow, and green arms, and they each had their own placebo. So you're randomizing to different potentially modes of administration of a placebo. So you might be worried about the differential effect of a placebo by its mode of administration. So within the trial, you might have a model that recognizes the potential heterogeneity of the placebo effect. So that can be an added feature to these trials. Another one is many of these platform trials continue to run, and after the first 3 arms, we add another arm and another arm and another arm, and we continue enroll controls over the course of the trial. That opens a new feature to these trials, which is time. Here is an example perpetual platform trial where when it started, there were 2 agents and a control. The control is represented by the black arm throughout, and then each of the individual agents is a different color. You can see they start and stop at different times throughout the course of the trial, and there are 12 of them. And the y-axis here is showing the relative response of the arms to control. Notice the control has different rates over time. Earlier on, the success rates were lower, near 20%, increasing up to near 45% as the time went on in the trial. So we can model this effect of control to make sure we have appropriate effects measured for each of the individual agents. So if we focus on only the control, you can see that there's heterogeneity over time. So in a trial, in a perpetual platform trial, we model the control over time. Here's an example of a modeling of that control, and then we can look at the relative effect of a treatment compared to this control where we've modeled time. And so this shows the example of the left with arms enrolled at different times. Relative to control, we can adjust through the statistical time machine to estimate each of the arms' relative efficacy on a same playing field. So this is a complete comparison, a one-to-one, apples-to-apples comparison, if you will, through the adjustment of time. So this allows perpetual platform trials. So platform trials can greatly decrease the amount of resources needed in trials. I've showed you examples of that where by sharing controls across arms, we can enroll as much as 67% less controls with the same or better inferences for each of the arms. It results in decreased time and resources for these trials, better, if not the same, inferences. Patients really like the fact that we're enrolling far fewer placebos or controls in trials. It brings about new statistical questions. How do we model time in trials and potentially different modes of administrations of these controls? It's well worth the effort in these trials for the added benefit. So thank you for your time. Thank you for watching.